But well, by way of introduction, I should say that I thought this was going to be a group of about 10 or 20 people speaking informally, and I thought this was an opportunity for me to read up stuff. Then I figured that it might well be the entire symphony, so I'm one of many speakers, but it turns out that that's going to be later. But nevertheless, this was a great opportunity for me to do some reading, and both about Darcy Thompson's book and also about patterning in biology. Now, I'll get started, but first of all, this is a book, and this is not the title of the book. The title of the book is on growth and form. I added function to it. So this is a book which everyone doing biology hears about when they're growing up doing biology. Many people buy it, nowadays probably get it online in some form or the other, and very few people actually read it. And at first reading, it seems a bit daunting. It's huge. It's a voluminous piece of work. It's hugely illustrated, and that's very nice. And I'll just get some water. It's not clear from today's biology, even from the point of view of biology of a few decades ago, what the message is, at least at first reading. But as you read it further, and what I did recently when I got Alok's mail was to buy it on Kindle. There is a abridged version abridged by J.T. Bonner, and he has condensed a 1,000-page book to about 780 pages or so, which is not much, but the important point is that it's also annotated, and that's nice. So if you want to read the book, then that's the version which you might want to read. But the book itself has three aspects to it. One, it looks at all forms in nature, both inorganic and organic. And the point it tries to make is that there are some key rules which underlie the nature of patterns we see. Now, that's not in itself a new statement. For thousands of years, maybe millennia, humans have tried to look at the nature of forms and what the underlying rules are. Typically, these attempts have looked at the skies, and from that we have learned about astronomy, about the planets, about the solar system, about the sun, and about the stars. And those mathematical laws have proven extraordinarily insightful and valuable even today. We've also had extraordinary similar insights in mathematics itself, looking at how patterns come about and the nature of these patterns and the rules in mathematics which underlie these patterns. And they have been, again, very valuable in terms of beauty and also in terms of the rules and the abstractions which are possible. And that's been one course of looking at patterns. But the patterns which we see around us in our day-to-day lives, such as mountains and seas and trees and birds and so on, how do they come about? And what Darcy Thompson's book tries to address is to see whether there are underlying rules which link patterns around us, both inorganic and organic. And the point which he would like to make, which is the first one, is indeed there are, and these principles are rather deep and will tell you what's the unifying theme which creates all these patterns. I'll come to what potentially creates these patterns and what his views were. The second point is his views on evolution. Around the time he first wrote the book, the first edition in 1917, evolution, the theory of evolution by natural selection, was in full acceptance. There was a view that everything could be explained in life by how it was adapted to be where it was. And it was this just so view which D'Arcy Thompson did not accept. And he wrote his book essentially without explicitly saying so, but it was an anti-Darwinian statement. He wrote to his publisher that that was his view, but he tried not to bring his view to bear on the book. The book was just about various kinds of patterns and what the rules which govern them, but not his views about how they came about to be. But he was clearly someone who did not think that natural selection was important, even if it operated at some level. What then causes patterns to come about? And he bordered on what we would, in the 20th century even, and even now, call a vitalist viewpoint, that there was some agent, therefore, which brought about this pattern in a very general manner across inorganic and life forms. So that was the second point. And the third aspect of the book was that the tempo of change. He believed that species didn't evolve gradually, as was suggested by Darwin's theory, but change came about in a saltatory manner, in jumps. So that again was a very hot debate some years ago in evolutionary biology, and still there are questions about the tempo of evolution which are not resolved and also are still a matter of debate. Now, one of the best ways for all of you, I would say, to get into the book is actually read a very nice commentary on the book. There are several. The preface by Stephen Jay Gould to Bonner's edition, I don't know whether it was independently written for that edition, but there's a very nice preface. That is very nice, very valuable to read. But Philip Ball, who writes about all sorts of things in science, in nature, he has written a very nice commentary on growth and form. It's called In Retrospect, and that was, I think, published in 2013, and that's a very valuable retrospective on the book itself. So what I will do is go through the book through the lens which Philip Ball provides. So I'll summarize his summary, and then I will give some examples from biology, which we've been doing over the last couple of decades, on how developmental biologists address the coming about of form and how it relates to function. Now, it's important in that context to keep in mind the kinds of questions which biologists ask. And Sidney Brenner famously said that they ask only three questions. One is, where does it come from? That's evolution. How is it made? That's developmental biology. And how does it work? That's physiology. So from that perspective, how do those kinds of approaches fall in place with D'Arcy Thompson's book? So one answer to that question comes from a very nice lecture, which again is something which you might want to look at, on patterns in biology by L. Mahadevan. He's a professor at Harvard and he visits here often. And if you Google that lecture, you will, I think it was given a few years ago, he addresses this issue in a very interesting manner. He says that physicists who are interested in living matter and who look at patterns in matter and look for unifying rules are basically looking at a top-down view of how patterns come about. And biologists really are coming with a bottom-up view. And it's very interesting to see how those both those views actually end up merging into a kind of a synthesis. And today, as we know a lot more about the molecular, cellular, developmental mechanisms which operate inside an animal and in shaping the outside of an animal, many of the heated debates of that time seem to be not so relevant, but are leading to exciting science. So let's go through what Philip Wall's commentary really says. First of all, he points out that in a manner similar to Newton's tome, the Principia Mathematica, Thomson's book is also not read. It's bought often, it's kept on your shelf, but it's not read. But yet there's, a very important point there in this book, which it was first written in 1917 when the Neo-Darwinian synthesis, that is, how genetics and population genetics come about to impact on evolution, hadn't yet fully taken place. But by the time the second edition came about in 1942, it was already in full swing, as it were, growing even stronger. And therefore, many of the points which the book, Darcy Thompson's book, brings about seem to be dated. Yet they have a lasting effect, the book, for reasons we'll come to later. Now, the key point in the book was summarized in the preface to the first edition by a famous statistician, Pearson, whose statistics many of you use. And he says that, to paraphrase that statement, that there will come a time when biologists will not hesitate to use mathematics in their work when they need it. And in many ways that time has come now, and we don't hesitate to use mathematics at all. And basically the mathematical principles as a shaping agency that may supersede natural selection, which was Thomson's view, is now being addressed by asking how these mathematical rules interdigitate with natural selection bottom up to see how form comes about. Thomson himself was an interesting character. Here he says that in a manner similar to what many ancient philosophers, Greece, India, Egypt, have talked about, is that the harmony of the world is made manifest in form and number, and the heart and soul and all the poetry of natural philosophy are embodied in the concept of mathematical beauty. So this statement underlies what his view was about everything, which is there has to be a rule which is beautiful, a mathematical rule, which is all-encompassing, which explains the rules of that form across all inorganic and organic matter. He grew up in Cambridge, then went, like Darwin, to study in Edinburgh medicine, left that, came back to Cambridge, and then went to Scotland again, to a relatively smaller university after first being in Dundee. He went to St. Andrews, and he was the chair of natural history there, and basically he hit the academic landscape around 1894 with a paper where he argued that physical forces and not heredity may govern biological form. So it's really how forces act on cells and organisms which govern form, and it's nothing to do with biology at all. And this needs to be sort of reconciled with a view about natural selection and how form comes about because of natural selection. So I'll now, take a short detour from Darcy Thompson's book to say how the phenomenon which led people to accept natural selection widely at that time is viewed today. And the first comes to mind are Darwin's finches. Now, Darwin's finches are distributed in the islands of the Galapagos, And depending on what's available on each island in terms of food, the beak shape is distinctive to allow the birds to live on those food, and therefore selective advantage allowed those birds with the appropriate beak size to survive and also speciate because they were isolated from birds in other islands. So that's the distribution you see of the finches in various islands. I've not shown the map where they are, but there are different kinds of beaks and beak sizes. Now, how could this have come about, and how would you explain this from the point of view of looking at patterning from a macro level? Of course, there are rules by which beaks, the pattern of beaks could be understood as in terms of the shape, in terms of the material they're made, in a manner which Darcy Thompson looked at other shapes, antlers and so on and so forth. And that those will be very valuable, and those will also put these beaks as, again, perfectly adapted to their environment. Today we know, largely from the work initiated first by Cliff Tabin's lab, what the molecular basis of this is. And it turns out that comes about by regulatory mutations in genes which affect the BMP signaling pathway and the, CAM kinase 2 pathway. So by altering the regulation of genes which are involved in the hierarchy of steps involved in beak design and beak formation, you can get rather dramatic changes in beak shape. And the question is, what is the time scale of these kinds of changes? And by looking at the kinds of mutations which are there, they're typically small changes in regulatory elements. They're not caused by leaps of changes caused by, say, transposable elements hopping into these regions. It looks like this has actually taken a significant amount of time. Similarly, you could look at scaling in another context, and that is the size of dogs. And there are very, very small dogs perfectly formed, and very large ones also perfectly formed. And these, again, will demand the answer about how can you have a similar shape growing in size, what are the rules. You can calculate what is the biggest size this shape can hold. Then you also have to calculate how internal tissues will pump blood and be intact as the size grows larger or smaller, metabolism, energy, and so on and so forth. And these approaches will tell you rules by which these sizes can scale. But from a bottom-up level, it turns out, again, that, if I'm right, I don't know, about 70 or 80% of the breeding, the molecular changes underlying the breeding of dogs for size, comes from changes in growth factor genes. And therefore, a very small number of genes can account, because of the regulatory changes, both in terms of size and length of dogs. And again, these come about by breeding over a very long time. So you can have a macro level view of how form and what the rules are. The bottom-up view also tells you about how it actually comes about. And we don't today see a contradiction in these two. We see that the laws of physics have to be observed, of course, but we see molecular mechanisms by which these growths can take place. And we'll come to a little later about how this has to be done in a manner which all internal tissues as well as external form have to be juxtaposed just correctly. Again, there are very large sizes to which animals can grow, and plants too. You have the sequoia trees, and you have giraffes over here, and their scaling, their growth of their neck requires an increase in the number of cervical vertebrae, and that we know today is caused by the action of Hox genes, which can expand or contract that number. And again, the question is, how is this limit set? Why is it a certain number and not more? After a certain number is set at birth, how does size grow? What sets the limits on size and so on? So these were the sort of questions on how growth takes place and what the limits on growth are, which Darcy Thompson addresses. And basically he says that in general, no organic form exists such as are in conformity with physical and mathematical laws. safe such as those which are in conformity with physical and mathematical laws. And his demonstration of this claim takes him through a whole range of topics. And these look at mathematics related to growth, flight, locomotion, and how mass and size, and these are areas which today, how tissue growth and size are regulated is a very active area, and how shapes of cells relate to shapes of bubbles and soap forms, the compartmentalization, the geometry of honeycombs, and so on and so forth. So these are very important broad pattern themes which he addresses, and he's most famous for his adoption of the logarithmic spiral, which is the central theme of his book. And there's a plaque commemorating him with this spiral at his residence in St. Andrews. And he saw this first in foraminifera, and then in seashells, in horns and claws, in plants, and so on and so forth, insect flight paths. And this to him was importantly, as Philip Ball says, evidence of the universality of form and the reduction of diverse phenomena to a few mathematical governing principles. So it was mathematics which governed how this happens, how growth and form come together. How does that relate today to our experimental sciences? And typically, what has happened in developmental biology is developmental biologists would have read D'Arcy Thompson's book, they pay homage to it, say it's a fantastic book, and then go on to study developmental biology their own way, right? And this is a figure from a paper by Lawrence et al., Peter Lawrence, Gary Strulin, Jose Casal, and they are trying to synthesize in that paper in Nature Genetics, I think it's in Nature Cell Biology in 2008, about how sizes of tissues are regulated. And they argue when you have a sheet of cells and you have each cell having a particular polarity, as you would if you look at, for example, the hair on one's arm, they all have pointed in a particular direction. How does this polarity come about? And this, they argue, and then how does the tissue, the sheet of cells, have a particular size and growth? So there's an intricate linkage between pattern and growth, and the genes and gene products involved in patterning are also somehow must be involved in limiting growth or regulating growth to the right extent. And basically they argue that there's a gradient of specific molecules, they talk about protocadherins in this case, across groups of cells, which set the steepness, a right-hand side and a left-hand side, and the lower limit sets a threshold beyond which growth cannot occur, and therefore you get the patterning, the scaling, as well as the growth limitation by this method. And this is, there's been a lot of work, which I won't go into now, using signaling pathways which are conserved, such as the Wnt signaling pathway, the BAP signaling pathway, and seeing how scaling and pattern formation both take place together. But yet, overall, the theme of D'Arcy Thompson's book was not to look at these kinds of mechanisms at all. He was reacting against Darwinism, which had a very strong positive impact on people all over Europe. And the Darwinist view was that it is sufficient, as I said, to account for every aspect of form that you see, and that could be explained by adaptation. So Thompson's arguments were more from an engineering perspective, and he did not challenge the idea outright that natural selection was used by nature, was nature's scalpel, as Philip Ball says, but he specifically argued that there were constraints imposed on what it could do given the laws of physics, as it were, and therefore the forms that might emerge. And that doesn't seem to be something which one needs to argue with. And as I said, when he sent the manuscript of his book to his publisher, he argued, however, about his strong anti-Darwinian views, even though he didn't explicitly leave that there. So his views on evolution were very strong. So the third point, one is about the form, the second is about evolution, the third aspect is about the tempo of evolution. And here's Stephen Jay Gould, and both Gould and his colleagues, Stephen Eldridge particularly, they proposed many years ago a view that evolution could also take place in huge leaps. And they argued that the gaps in the fossil record substantiated that view. And there's a lot of controversy about that, whether the gaps are due to the gaps of fossils not being found, and the cases where they have been found show more non-saltatory view of evolution. And that debate went on for a while. And again, by looking at molecular markers of change, it's not clear that saltatory changes are the ones which are dominant, if at all they are there in some specific cases. I mean, one can imagine, and Gould himself has pointed this out in one of his books, I don't remember which one, that there's no rule that evolution must act slowly. If, for example, a lethal virus were to infect this room and a small number of people had mutations in genes which allowed them to be resistant to it, whereas the entire population were susceptible, evolution could act in an instant and cause that lethality. So it's not necessary that evolution must act selection can act right away, and therefore that combined with isolation could result in a variety of consequences which are rather rapid, depending on what the kinds of genes are which are involved. So it's not just that evolution, that selection can act rapidly, but whether selection acting rapidly can have consequences on speciation, and that's a big debate which is still going on, particularly in more complex life forms. So there are, of course, lots of such open questions about evolution and the deterministic nature of the course of evolution, and therefore that's a great value in this book, that despite its contentious nature about D'Arcy Thompson's views on evolution and the nature of his views on saltation, there is still great value in the book. Now, another major value in the book is the importance of how pattern organization takes place. And self-organization is something which we see in a variety of natural phenomena, and how that comes about is very, very exciting and interesting, and what are the physical rules for that. And you see this again in both inorganic and organic contexts, for example, in animal swarming, in how different populations auto-regulate themselves, the way crowds move when they go out of an auditorium or come in. And the rules of this are something which are very interesting, mathematics and physics, and it's again being examined a lot. And therefore, observing the kinds of patterns is something very important to be able to see how actually they come about. Mahadevan's lecture, which I referred to, talks about something which at first glance seems rather gory, which is how our intestines are patterned. And he shows how a way by which you could exert pressures, but also have cell division in a polarized manner with tip cells, or different layers of cells having different properties, can have dramatic effects both in intestinal patterning, but he also talks about tube patterning in plants. So there are common principles underlying the way tubes are made, which one can get by these kinds of mathematics, and these apply not only to living matter, but others as well. So this, though, about this aspect about Thompson's work, about self-organizing being a very interesting aspect of study, which it is now, he just pays sort of lip service to that, and he says it's self-evident what the chemical mechanisms are, but he doesn't elaborate on that. and therefore to use him as Philip Ball says, a prophet of self-organizing systems would be incorrect. Yet the fact that many of the systems he looked at are now being studied for their self-organizing properties is certainly true. So, for example, the aspects he looked at, such as the cracked polygonal networks which you see when mud dries up, the stripe marking of animals, the other aspects such as how shapes in human face take place and patterning comes about. These are aspects which are studied not just from the self-organizing perspective but patterning, and that was actually the mechanisms underlying it, the chemistry underlying those was something which was articulated a little later by Alan Turing. And Turing looked at basically a circle of cells, a donut of cells, and asked how you could get patterns. He showed that there were rules by which you could get multiple kinds of stable patterns coming up, and by proposing activators and inhibitors, he suggested that these mechanisms could explain how patterns came about in biological systems such as fish and zebra and so on and so forth. Now, those mechanisms too don't have many examples in biological patterning today. There are few, some of them are significant, but there are very few examples of Turing patterning in biology, but that was at least an attempt to bring chemistry into patterning, which D'Arcy Thompson very carefully did not address. Now, there were people who looked at patterning, such as I mentioned before, mathematicians who looked at more abstract aspects of patterning. But D'Arcy Thompson was inspired more by biologists, anatomists, morphologists who looked at patterning, and in particular, he was inspired by two people, Wilhelm His, and that's over there, and Wilhelm His was the person who invented the microtome. He showed how one could fix samples and then slice them in a manner where you could then reconstruct their three-dimensional shapes, which we do now in really fancy ways. And that was very important for looking at patterns in various tissues, such as the heart and a variety of internal organs, and also external patterns, of course. Willem Roos here was another person whose work Thompson looked at, and Roos was again trying to look at how patterns came about in biology, more by looking at external developments, such as you see in amphibians. And what he did was to take an amphibian embryo at a two-cell stage, ablate one cell, and ask whether you got half an animal on one side, which was not ablated, and indeed one did. And this led him to think that there was a mechanistic rule by which patterning takes place, and the two-cell embryo, of course, therefore knew, quote unquote, that which half each cell had to make. It turned out that this experiment was repeated in another context by Hans Driesch in Trieste with sea urchin, and there Driesch took apart the two cells at the two-cell stage using a hair and let them on a Petri dish overnight, and showed later on that each cell could actually make an entire sea urchin. And these resulted in two very different views of biology. Willem Roos' experiment, interestingly, for someone whose work inspired D'Arcy Thompson, suggested that there were components which were divided between the two cells, and there were rules internal which decided which cell would make which half of the animal. So there was a very material explanation for this. Driesch came to the conclusion, the fact that each separate cell could make an entire organism, that actually there was a vital force from outside which knew that this other cell was not there, and therefore a new organism had to be made. So those were the kinds of contradictions. And again, we don't have too much time, though I'll mention it a little bit later about what the mechanisms are for these kinds of differences. But these were the kinds of people, rather than mathematicians, who inspired D'Arcy Thompson. Now, this aspect about how the biology of cells interacting with each other, and the biophysics of that interaction, how cell-cell adhesion molecules cause cells to change shape, size, their migratory properties, all of that is a very, very exciting field which now today combines the study of the physics of intercellular interactions with the biology and the consequences. And it's a big problem because it's not easy at all to dissect out what contributions each component has. But there are extremely interesting tools now which allow one to deform cells, change their adhesive properties, and look at the consequences on other properties of the cells, including gene expression. So, why then, if in terms of patterning, if D'Arcy Thompson has told us in many ways today what we see as obvious, he's written it in a beautiful language, he's written it in the language of, people who know their classics well, and, these are polymaths who know music, they know art, they know languages, they look at form, they know mathematics. It's a wonderful read, but it doesn't tell us anything about mechanisms. Its links to evolution, we would say today, are not valid, and its links to the mechanisms of how these forms come about are absent. And why then, therefore, would we value such a book? Now, much has been written about it, and particularly I would urge you to read work by people such as Philip Ball himself, Peter Medawar, Stephen Jay Gould, J.T. Bonner. But his breadth of scholarship is, of course, amazing, but it's also a separate kind of value today. Today, we find very, very few people who are able to look at science across the spectrum, physics, chemistry, mathematics, biology, engineering, and write something about aspects which encompass all these fields, or answer questions which encompass all these fields, not looking at them as a synthesis of these fields, but addressing a big, broad question. And these people are, extraordinarily daring, some would say foolhardy people, but they're extremely valuable at a time when much of our work today focuses on, putting our nose to the ground and making sure that we get specific things done. We forget the big picture and looking at the details. The details are important, but these are very few people, such as Lynn Margulis, very pixelated here. She first proposed the idea of symbiosis in evolution, about how mitochondria could have come upon to eukaryotes by symbiosis. And James Lovelock here proposes the idea of how the entire planet is one organism, as it were. And these are ideas which, it's easy to nitpick and shoot down in one way or the other, but these are also great intellectuals who challenge us to think in extraordinarily interesting manner. So these are the reasons, in an era of citation counting and tenure battles and funding crisis, as Philip Ball says, these are very, very important people, and one shouldn't forget to celebrate them and read them, and it's very good that you're doing that here. Now let's go to how the bottom-up view is, and that's something which we do all the time, and we can ask how, from that view, these patterns come about. You can look at wings of birds, insects, flies, and look at it from a mechanistic point of view from outside, and that's absolutely invaluable. But how does this so-called just so adaptation come about? And fortunately, because of a dramatic revolution in both molecular biology and in developmental biology, and those coming together through developmental genetics, we've got many of the tools which can actually bridge the gap between gene expression, cellular function, cell biology, and phenotype. And that's a very important bottom-up complement to such important top-down views. And of course, the organism which was studied hugely for these kinds of aspects turned out, again by a series of accidents, to be the fruit fly. And it's important that in our own context, at the TIFR Molecular Biology Unit in then Bombay, now Mumbai, P. Babu started studying how the wing of the fly is made, and a gene which affects wing development, wingless, which was discovered at the Indian Agricultural Research Institute by Sharma and Chopra, was a gene he used to study. He studied in Bombay, and Peter Lawrence studied in Cambridge at the same time. And that was a time when they used to compare results with each other, and those they used to do by sending telex messages to each other. Babu would write to Peter Lawrence saying, "Wingless, not cell autonomous, stop," and which Peter Lawrence would write back saying, "But is it tissue autonomous, stop?" Yes, stop, So those are the kinds of, and the telex operator, of course, you have to go and fill in a form and give the, message there, and she would type it out, it would go there, depending on the time zone difference. So every day there used to be a, paper torn out, really old kind of paper, which is a carbon copy, which was kept by the operator in case you're getting nasty messages. So she would have proof of your interactions. Everything had to be official. You couldn't send non-official messages like, "Can I come and party in Cambridge?" Stop. It had to be all official work. So it turned out that it was very interesting that wingless was one of the genes which was involved in patterning the wing, but the general idea which came about, and this came about by a comprehensive study of patterning, literally bottom up, by Jari Niswain-Pollard and Eric Wieschaus at the European Molecular Biology Laboratory, where they were postdocs. And they asked a simple question, not top down, what are the rules which make pattern, but bottom up, asking, what are the genes which, when mutated, result in an alteration of pattern? They were not looking just for messed up pattern. They were not looking for genes which affected pattern by messing up the animal. They asked, are there genes which will affect pattern in interesting ways, which give us an insight to how the animal is made? Now, it turns out that it was extraordinarily fortuitous on two counts. One, that Drosophila develops early on by its fertilized egg dividing without forming new cells. So there's nuclear division and no cell division. And so there's a syncytium. And then it is fortuitous that the genes which are involved in patterning turned out to be transcription factors. And these transcription factors were expressed in a pattern which gave you insights into pattern. And if, for example, such a screen had been done in zebrafish, if hypothetically one could do such a screen, you would have got a very, very different kind of result. But it was extraordinarily beautiful because you saw at the blastoderm stage, at the nuclear blastoderm stage, at the cellular blastoderm stage, the expression patterns, then by RNA in situ, later on by antibodies raised to gene products, and later on by tagged genes whose expression patterns we can see now dynamically, that genes are transcribed in a manner which tell us about how patterning takes place, and their localization tells us how head, thorax, abdomen, and tail are patterned. Right? And this was absolutely extraordinary because it allowed us to bridge this phenotype-genotype gap, not just by making mutations and looking at the consequences on the phenotype of the animal, but by seeing how gene expression patterns lead to that phenotype. And that was absolutely astounding. So just as we learned a lot about how patterning in the embryo could take place, and how the wonderful pattern of the maggot was the rules underlying that, once those genes were cloned in flies, and once it was possible to clone homologs in other animals, in mouse and human, then it turns out that these genes, many of them are actually conserved and involved in patterning humans and other animals also. So the cervical vertebrae of giraffe, for example, the numbers of vertebrae would be determined by the number of the actional specific combination of Hox genes, and similarly in humans, similarly the axial patterning is also determined by Hox genes in insects and mice and so on. Now, the patterning of external structures, such as the thorax and the wings, which you see here, takes place on what's essentially a sheet of cells. And the broad principle is that this sheet starts from a small number of cells, let's hypothetically say as 4, and the first event in patterning is compartmentalizing these 4 into, let's say, 2 on one side and 2 on the other. So here's your sheet of cells. You divide this into the small number, and you have a rule, a transcription factor, which says this is different from that. This transcription factor then sets up a expression of a new molecule on this side, which has a short-range signaling, right, which patterns the molecules on the other side, and also a long-range signaling, which acts on both sides over a very long range. And because there's a transcription factor expressed on one side and not on the other, the consequences of this expression of this long-range transcription factor and also the short-range one are different on both sides. And this is sort of a general rule which Peter Lawrence proposed many years ago, and still by and large valid. That is, what you do is you divide cells into compartments, you set up gradients which can act on both sides in differential manner, and you do this again and again. And if you do this again and again, and you have restrictions on which genes can act where, on which target genes, then you can use the same kinds of factors again and again to pattern external tissues. Now, both Darcy Thompson and these kinds of studies, famously by Lewis Wolpert on the French flag problem as a solution for patterning, and by people such as Peter Lawrence and others, look largely at external tissues and their patterning. But Lawrence himself pointed out, as have several others, that we also need to have below this the patterning of tissues so that this pattern outside can lead to function. And that's something which, for example, can be done by looking at muscles in the fly. What are the rules for, even from a bottom-up perspective, for looking at how internal tissues such as muscles are patterned? And that is something which turns out to be very different, but also conserved in many animals. And that is that the outside, the notum, the thorax, signals to cells in a similar manner, muscle progenitor cells, well before they have reached the right population size, about how to divide, how much to divide, and where to be placed underlying the presumptive thorax. And then, as differentiation takes place during metamorphosis, these cells differentiate according to, again, similar rules, using the same kinds of molecules to fit this correct position in the thorax. So 3 things have to come in place. The right number of muscle progenitors have to divide, and those rules are dictated by and large by the epidermis, with some intrinsic properties of the muscle themselves. The muscle, as they develop, the fibers need to attach to the right places, and they need to also be innervated correctly. They also need to be different, even though different segments. The thorax shown there, and a little green bit you see in the abdomen, the numbers of progenitors set aside in the embryo to make these kinds of muscles are not very different. Yet, during larval development, the progenitors of muscles in the thorax need to divide a lot more than those in the abdomen, and they need to make muscles of very different sizes. So what are the rules which determine both intrinsic and external, the sizes of the muscles? Are they similar to those kind of sheath cells and the protocaderants which Lawrence talked about in determining the size, or there are other rules operating? And that's something which has been addressed by many people, Christoph Jagla's group, for example, has looked at muscles in the embryo. This is a picture of muscles in the adult abdomen, not in the embryo. But basically, what the conclusion there is that there are specific important nuclei. Muscles are multinucleate, and some of the nuclei are special, such as those shown in red here. And they, depending on where they are on the axis of the embryo, have the property, because of their Hox gene expression and other combinatorial transcription factors, to say when they will stop the muscle from growing. But that is also a context-dependent phenomenon, depending on the attachment sites. So that combination of muscle intrinsic properties and where it attaches and how many cells are fused to this muscle determines the muscle size. And in the thorax, we think that it's very different. The muscle attachment sites are much, much larger, and shown in red over here, and the green is the growing muscle. And we think that this interaction between the muscle attachment site on a much larger scale and the growing muscle and a very much larger subset of myoblasts sets what the limits on size has to be. And similarly, how much branching in the innervation is needed and when to stop is a feedback interaction between growing muscle and growing nervous system. So by these kinds of rules, you end up making a muscle under the thorax, which is very different from the muscle in the neck segment, where there's a structure called the haltere, and where, again, the same number of cells are set aside during embryonic development for the wing and the haltere, yet the haltere is very, very different in shape and size and form and function from the wing. Now, there is one gene called ultrabithorax, which if you remove specifically in the third thoracic segment, the second thoracic segment is where the wings are, and the third thoracic segment is where this little structure called the haltere is. If you remove UBX from the third thoracic segment, you can transform the third thoracic segment to the second thoracic segment. And this was work shown beautifully by Ed Lewis, and he got his Nobel Prize for this in part. Is this dramatic transformation evolutionarily significant? Could this be a way by which you could have saltatory changes, by which you could get four-winged flies, insects going around? It turns out, after much investigation, that that is not the case. That in four-winged insects, it's not by removal of UBX in T3 that you get a transformed T2, but the targets of UBX are different, right? And evolutionarily, that also seems to have taken place in a manner which is different from this. So Ed Lewis's original view that homeotic transformations are atavistic, they take you back in time, doesn't seem to be founded, and he himself changed that view hugely. He found another set of mutations which was a gain of function, where the abdominal pigmentation could be seen in the thorax, and therefore it seemed as if a new abdomen was being created. This was because of the misexpression of an abdominal regulatory gene, ABDB, in the thorax. But that, it turned out, was because the ABDB gene was rearranged to be close to the promoter of the muscle attachment gene, and therefore showed an abdominal pattern similar to that of muscle attachment. So these kinds of changes induced by mutations cause dramatic changes, but none of them seem to be in a manner also such as the Antennapedia mutation, which tell us about saltatory action evolutionarily. Does this four-winged fly have muscles inside the transformed thorax? The answer is no. Because, as I said, you need both muscle intrinsic properties and properties from the notum to make that transformation. And this fly transforms only the external aspects of patterning and not the muscle aspects, and therefore you don't see that. And now what Dev Kanyadatta and Uma Shankar have shown in the lab is by making those changes in the muscle too in a very different way, you can start getting muscles in the four-winged fly. So you can, in principle, construct a four-winged fly with muscle, but it's in very bad shape, and it's in no way similar to four-winged insects at all. which have a very different way of using UBX genes in the third thoracic segment. If this is the way, the interaction is there between external structures and internal ones, how is it for structures such as the brain? And I'll end with that. Our brain, and perhaps the brain's, sorry, by our brain I mean the fly's brain, I sort of use that interchangeably. Habit for many years. Yeah. The fly's brain is made up by groups of stem cells set aside which have specific functional properties. So stem cells not only divide to give rise to other cells and more stem cells, but these populations which arise, neurons or other cell types, from a stem cell and its lineage are often functionally similar within a lineage. So stem cell lineages are not only tell us about how the anatomy of a brain comes about, and these color codes are different stem cell lineages, but they also tell us about the function of these kinds of lineages. So you have, for example, you can have all of a particular glutamatergic neuronal lineage coming from one particular stem cell lineage in a particular part of the brain. Just as you can get dramatic switches in external structures by making changes in a single gene, you can get dramatic switches in the brain and their patterning by making changes in a single gene. So there is a gene called OTD, orthodentical, which normally is expressed in a particular stem cell lineage in the fly's brain and results in the neurons going to a region called the central complex. The removal of OTD specifically in that lineage will transform this entire lineage into one which innervates the anterior lobe. So just by one switch, you have moved a central complex lineage to innervating an anterior lobe. But not only is this, not only does this happen, but unlike the case of the four-winged fly, this lineage is functional. It can now smell, and Sonia Sen has shown this beautifully, both in behavioral assays and also in physiology. And it also smells the right kinds of smells as expected for this kind of a new population of neurons. So can one, therefore, pattern the brain by making these kinds of switches, and do these switches happen very rapidly? And that's a very interesting question for the evolution of patterning of brains such as ours, and now I mean human brains, where the numbers of neurons are huge, and the numbers of neurons in specific regions, cortical areas, are much larger than other primates. And how did this expansion of neuronal number come about? Using the same mechanisms which are present in our non-human primate cousins. Could it be because of an amplification of stem cell lineages? And that's something which we don't know the answer. But if it is, it would be interesting to see whether this came about in a relatively gradual manner or a saltatory manner. The energy required to create a brain of the size we have is huge, and is possible only if we stop eating raw food 24/7, but start cooking food and eating that. That's one view. So the energy for brain development could come from cooked food, but how does that translate mechanistically to altered brain development? That's something we don't know, whether stem cells are involved, whether there was a rapid change as we in our brain size, or whether it was slow, and if it was whatever it was, how is that developmentally feasible? And that's something which I think we have the tools today in model organisms to try and investigate. So to end, we can now look, unlike the time when Darcy Thompson was looking, at patterning not just in the outside world, but across scale in biology, right from cellular interactions to organisms and ecosystems. The rules, the general rules of any for these kinds of patterns, we don't know. People such as Peter Lawrence argue that there would be general rules, such as the steepness of gradients. The kinds of molecules would vary from context to context. But the rules that you somehow measure the concentration of something in your neighboring cell, and then decide how to act, and the polarity and the growth is a consequence of that, and there are thresholds which limit that growth, is suggested by him to be a very general mechanism by which both growth and form are integrated, and function comes about by contact of cells with each other, so that functional feedback can also come back into this. Thank you. Questions? I get very nervous when I see Sanjay in the audience. Yeah, sure.
So, taking the top-down and bottom-up perspective, are there specific examples where are there specific examples where there are contradictions in terms of applying largely physical or mathematical principles versus more bottom-up biology principles? Are there examples where it falls apart, or they should be complementary? But
yeah, I don't know whether there's any statement in the top-down which is flawed in itself. It doesn't. Darcy Thompson's approach doesn't address mechanisms specifically, but it is completely consistent with the laws of nature, as it were. And the bottom-up also has to be. It's just the bottom-up is, of course, messy. It's a series of accidents, and whose mechanisms vary from organism to organism. The chemicals used may vary, but it follows the rules of the top-down. Yeah.
How do you think the bottom-up or the finding out of mechanisms is really informed by the top-down approach, or the constraints on or the mechanism by which the whole system can work? So, do we really make use of that information in figuring out the mechanism?
Well, the problem is that even those who start out that way look at a broader question to ask, and then hunker down and start looking at a specific context, are faced with the reality that biology is so poorly explored that the discovery of new treasures distracts us, and we get caught in the details of what's happening in the system we're looking at, and many people make extraordinary discoveries. So while there might be a superficial obeisance to a top-down view of understanding this or that, the bottom-up actually gets the details sorted out. I think the two examples, one is Yanni Nusslein-Pollard and Eric Wischer's did ask this question about patterning, and then it did an extraordinary job. That's an example of how top-down and bottom-up actually was linked very beautifully. But Ed Lewis's contribution to how Hox genes pattern the animal was asking a completely different question, which was about the gene. How can you use genetics to look at the fine structure of the gene? Seymour Benzer looked at the R2 phage, the R2 gene fine structure, and Ed Lewis looked at the bithorax complex. And both of them were making finer and finer recombinants, intragenic, to look at the nature of the gene. Ed Lewis didn't look at the animal at all for a very long time. And then when he looked at it, he found these patterning changes which were extraordinarily valuable. So that link came a little later. So my view is that this approach, which doesn't exclude other approaches, often involves exploration in completely uncharted areas, and then sometimes mechanisms come about and sometimes they don't. Darwin is an example of someone who was inspired by possible mechanisms in geology, but really didn't get into mechanisms in operating in evolution at all, whereas Mendel sort of did. Sanjay?
Yeah, so, Alan Turing's work is a really good example of case where you had a mathematical theory
Absolutely.
that suggested a underlying mechanism,
Yeah.
which you don't see that much in Darcy Thompson's
No.
book.
Yeah.
Are there other good examples of mathematical theories that have suggested to biologists that you should go look for something like this, and then found it and actually taken it to connect with the mathematical theory itself?
Well, Hans Meinhardt's work on patterning was extraordinarily valuable in looking at how gradients are related to pattern formation. Lewis Wolpert, even though he was partly experimentally inclined, but very little actually, his theory on how patterning takes place, the French flag problem, is an example of how theory has impacted hugely. Francis Crick and Peter Lawrence amplified on the work of Gines Morata, Pedro Ripoll, and Antonio Garcia Bellido and others about the compartment hypothesis, that cells are compartmentalized, and that compartmentalization, anterior, posterior, dorsoventral, proximal, distal, is critical. And that came from experimental work, but Crick and Lawrence took that and made it into a general theory. So there are examples. Many of these theories, as theories are, have flaws in them with hindsight, but they allow you to explore in a manner which is very valuable.
There are no other questions?
Thank you very much.
I'd like to make an announcement. So we are still preparing for the 14th. So those who are interested can still participate. We meet every Tuesday in the evening at 4. So if you are interested in participating and you're interested in these kind of topics and questions, please let me know. And
Can I just add one point? Actually, there are 3 people who would be very valuable for you to draw in for discussions, who have great depth and understanding in these areas. All 3 of them are close to Bangalore. Mahadevan, who I mentioned, then Sriram Ramaswamy, and Ajay Sood. There are others, but these 3 are close by, and they would be really terrific for these kinds of topics.
thank you. Thank you for coming on Saturday evening.